Um, our, our last speaker for the session and probably for the conference is going to talk to us about potentialities of bioinformatically predicted linear B-cell epitopes on dengue PRM protein by Nadugala, PH Premaratne, and CL Goonasekara. Thank you.
Good evening, everybody. I'm Nadugala from Kothalawala Defense University. I'm going to present the study on potentialities of bioinformatically predicted linear B-cell epitopes on the dengue PRM protein or the dengue pre-membrane protein. Okay, the objective of the study, first I will tell the objective of the study: to predict B-cell epitopes from DNV PRM membrane by bioinformatic tools. Actually, this is a different approach from the traditional research, because they used to move from antiserum to then identify the epitopes, then characterize those epitopes. So we have changed our approach, and we are moving from bioinformatics to laboratory research. This is my initial part of my PhD study under supervision of Dr. Charita Goonasekara and Dr. Prasad Premaratne. The main project is to develop a vaccine candidate with a broadly reactive neutralizing immunoresponse. Actually, theory made my life easy, because why we selected dengue, it has become a global burden. Annual cases of dengue is around 390 million cases worldwide. That is all dengue cases, not only hemorrhagic, all dengue cases. And we are in the region where about 0.5 million annual cases. Sri Lanka encountered the epidemic in 2009, but in dengue, as my final goal is to identify a leading vaccine candidate, I want to go through some leading approaches in vaccine research. The main one is live attenuated DNA vaccine, and there are approaches to find inactivate DB vaccines, etc. Our one is different from this approach, but I am not going to disclose at this moment. It will be disclosed once we succeed in that. Okay. As we know, dengue is an RNA virus, single-strand RNA virus. The genome is around 10.7 kilobases. There are 4 serotypes, DNA 1, 2, 3, and 4. That is the reason, as you all know, there's no vaccine that has been successful for the dengue. Only thing we have is the management practice of control of vector. So there are 4 serotypes genetically different. We are in our study, in the part of this study, we are concerned about the pre-membrane protein, which is the outer part of the DNV envelope, with the which is about 166 amino acids, and the PRM protein acts as the chaperone of correct folding and assembly of the envelope. The maturation or the infectivity of DNV depends on the cleavage of PRM protein to membrane protein. We are talking about in our study we are targeting on the B-cell epitopes, which are identification of B-cell epitopes is important in immunodetection as well as immunotherapeutic application. What is an epitope? Epitope is a minimal immune unit strong enough to elicit a potential humoral immune response with no harmful side effect to human body. Methodology, actually we started with bioinformatics, so we retrieved PRM sequences from NCBI, 50 sequences to each serotype, altogether 200 sequences for serotypes, and they were aligned on Mega 6 using Crustal W. And B-cell prediction was done using 3 tools: BabyPred, ELIPRO, SVM trip. These tools were selected after careful analysis of available web-based method and after consultation of expertise in these areas. Considering the model they have used to develop these bioinformatic programs, all are web-based methods, and also the characteristics they have used to predict the B-cell epitopes. After prediction of epitopes, the conservancy was measured, conservancy of epitopes within the serotype as well as among the 4 serotypes was measured, because always the problem with dengue was the genetical variation between the serotypes. And we also the conservation was visualized on web logo, and phylogenetic trees were constructed to each epitope that we develop. These are the results. As you can see, the different methods are given in different colors. There were the ELIPRO results, the prediction of epitopes predicted by ELIPRO method is given by, it was pink, I don't know now which the color has changed. Then baby pred, it's blue, and yellow SVM trip. Interestingly, most of the, after done the prediction, we observed that the tools predict the same site, almost same sites in the PRM protein. Here you can see one epitope was predicted, the same location was predicted by 2 methods, ELIPRO and baby pred. Results in summary: 12 epitopes were predicted for pre-membrane baby pred, 4 epitopes, then 6 epitopes by ELIPRO method, and 2 epitopes by SVM trip. The overall conservancy, that means the conservancy amongst 4 serotypes was around 50 to 70%, except some epitopes. And when we align the sequences of 4 serotypes, we saw that DNV 3 and 4 are genetically more similar than isolates of DNV 1 and DNV 2. Since also we observed that because of that, DNV 3 and DNV 4, that is the 2 serotypes, have higher, the epitopes have a higher serotypic conservancy, more than 80. Regarding the phylogenetic analysis, when we develop the phylogenetic trees for each epitope, no specific phylogenetic pattern or no geographical variation pattern was observed. I forgot to tell that when we are taking the isolate, the 50 isolates were selected from different geographical variation representing all over the world, as well as the temporal difference from 1963 to recent years. We downloaded the isolates. According to WebLogo results, we observed a partial conserved region from 65 to 170 amino acid region. Conclusion: this is a bioinformatics study. Actually, we identify it is a good initial step to screen potential linear B cell epitopes in dengue protein, and also as the studies started with antiserum and research-based studies, most of the epitopes were it showed that it can be potential serotypic, potential infectious enhancing epitopes, because there was a sequence difference in the predicted epitope. Also, EP9 and EP10 are potential vaccine candidates because they showed a higher overall conservancy among 4 serotypes, but we need to do some laboratory testing. And peptide stretch 55 to 65 can be used as a serotypic diagnostic marker because it showed a higher conservancy within serotype of DNV2, DNV3 and DNV4. These are my references. I would like to acknowledge National Science Foundation of Sri Lanka for granting for giving us the financial support, and these scientists for continuous guidance and advice. Thank you.
Thank you. Okay, I think.
Yeah, I think this is a very good example where you can use computational techniques to narrow down the, sort of like a mode of action. Like once they find out their laboratory data, they can go back and analyze, look at this data and then find a good mechanism. So, just to explain, why they are seeing their results. So basically now, with these computational models, you should be able to have 80% or so accuracy, and when you get the practical results, so that will show the right pathway to carry out research in the future in Sri Lanka. So this project is very well done, and I think the way you are going is quite nice, and what we want to see now is the practical part of it. So hopefully you will get the good practical results which you can compare with the computational models and show basically what is the accuracy that you have with the bioinformatic data actually. So very good.
Yeah, actually if I add something, we did check with these predictions were checked with IDB resources, that is the biggest database on epitopes. Some epitopes were, there were some assay-based results on the predictions.
Any more questions or comments for that matter?
Linear means the epitope, the activity is dependent on the sequence, not the conformation. There are two types of B-cell epitopes, linear epitopes and conformational B-cell epitopes. That is the sequence. That's why we can bioinformatically predict by these tools. If it is conformational epitopes, we have to go for 3D modeling and use those data for epitope prediction.
If I clarify what Nandan was asking, Dr. Nandan, what you are asking, a continuous epitope would be a sequence, a stretch of peptide which is continuous. So a conformational one is like once you have the 3D structure. So yeah, it's a discontinuous epitope, but when you have the whole model, then you can, the different parts are coming as the epitope, so it's not going to continue.
I have a question. So you compare different strains within Sri Lanka and across the range, is that what you said?
Yeah.
So Sri Lanka has 4 strains, and of that 2 are more closely related? Is that what you said?
3 and 4.
3 and 4 are more closely related?
Yeah, not only Sri Lanka, worldwide.
Okay.
By considering all the isolates I downloaded.
So you use which area of the genome you use?
That is the pre-membrane.
Oh, I see. So, and then you said that no phylogenetic structure.
No, not
it's
phylogenetic.
In the slide, that's how it was written.
No, it was like within the epitope, like we could not observe any phylogenetic pattern, like distinct phylogenetic pattern.
In
according
one strain
to the
or all strains?
All strains.
I thought it's still to me confounding what you're saying now and what you said earlier. So you have certain strains are similar and certain strains are not, right? 3 and 4.
3
similar.
and 4 within the serotype.
Yeah.
They are genetically more closely related.
Closely related.
Yeah.
And then, but when you are looking at the phylogenetic relatedness, they are similar, is what I said?
No, actually we were like expecting different geographical patterns within the epitope.
That's what I thought, yes.
But we could not.
Ah, I see.
That's it.
Okay, that makes sense.
Is it clear?
Yes, but if you look at, if you get them all together, I assume that each strain would have its own
Cluster.
Cluster.
Yeah.
Okay.
Yeah,
I see. Right. And then how these different clusters play out in the Sri Lankan map? Like are they found in different are these strains found in different areas in the country, or are they all together in Colombo, for example? How they, because that would be key to one path would be to identify which strains are more virulent, or which strains are more causing these the severe phases or cases, and or are they just they are so variable within, so that that would be if the viruses, if these, even though they have different phylogenetic history, if they are equally variable, then that would indicate the difficulty of going to actual vaccine or to any other treatment. So, or the other side is that there might be certain strains or certain types are easy to deal with than the other ones in terms of the variability of expression. So my question is, are there geographic structure?
Geographic structure means, like if you take Sri Lankan strains, they are in one cluster. Because they have not mentioned the specific location of the isolate. Sri Lanka just mentioned the country. So they are in a particular cluster. No. Only the country has been mentioned.
And also, I think even within countries, you find all those 4 strains. Yeah. It's not. I don't think that there is geographical preference. Yeah. It's widely. All 4 is widely spread, I think.
Not all
with my
4.
With my limited.
Not all 4, sir.
Not for all 4.
2 and 4.
2 and 4.
Yeah.
So are there countries that you only find 2 and 4?
No, in Sri Lanka I am telling.
Sri Lanka.
2 and 4 means mainly 2 and 4.
So you don't find 3 and
we have, but limited.
Yeah, that's what I said. Yeah. So is that ratio common right throughout the world?
That we have not studied.
That must be in the literature. I'm pretty sure. Okay, if there is no other questions, I'd like to thank the speaker.